Hi everyone, I'm Watchara Arthan. You can call me Krich. I'm from University of Reading and Royal Botanic Gardens, Kew. Today I'd like to present evolution of lineages, niches, and traits in the dominant savanna grasses Heteropogon and Thymida. Heteropogon and Thymida are silvergrass lineage distributed throughout the tropics, but Heteropogon and Thymida species occupy unequal distributions. The genera include wide-ranging species, Heteropogon contortus, which is pantropical species, and Thymida trianda, which is distributed in oval tropics. The distribution ranges of some species are more restricted. For example, Southeast Asia species, Thymida arundinacea, and Thymida caudata, and also endemic species in Himalayan mountain ranges, Thymida anathera. They also exist in diverse habitats throughout the tropics. Furthermore, phenotypes such as height, leaf size, on types, and on length vary within these species. The diversity in habitat preferences and traits draw questions around adaptations of species in the habitat and evolution of lineages related to habitat and traits. Their distribution ranges cover silvergrass ecosystem throughout the tropics. The silvergrass ecosystem are not just homogeneous habitat, but comprise of a variety of habitats. These habitats vary from very mesic ends in wetlands and floodplain, and mesic savannas such as pine savanna and tallgrass savanna, to very dry savannas such as acacia savanna in Africa. These habitats are controlled by different precipitation and temperature regimes, which influence species assemblage, species life history, functional traits, and also determine species distributions. Savanna and wetlands are two contrasting habitats in terms of bioclimatic regulating factors, dynamics, and species compositions. Savanna is generally governed by wet-dry cycle. Dry season usually lasts 4 to 7 months, and plants have to cope with drought and occasional fire, and then regrow in wet seasons. In contrast, wetlands are in inundated conditions all year round, but sometimes dry season could occur. In such conditions, plants face the problems with water saturation from standing water, competition to grow with other species, and anoxic conditions. These are common morphological traits that savanna and wetland species use to respond to different hydrological regimes. Wetland species live in the conditions with the less limited water supply than savanna species, so wetland species should grow rapidly and have greater height than savanna species existing in the dry conditions. Wetland species are expected to produce stewed root in order to support the structure. Increasing leaf length should be observed as these support plant production via photosynthesis. In addition, on types differently reacting to fire intensity, moisture level, and soil characteristics are hypothesized to be different between savanna and wetland species. So we first explore the phylogenetic patterns and relationships in Heteropogon and Thymida before we delve into further evolutionary and ecological aspects. Then we explore the habitat preferences of Heteropogon and Thymida lineages to see how these habitats have been changed and selected throughout the lineage. And then we define and differentiate the characteristics of bioclimatic niches of Heteropogon and Thymida species in savannas and wetlands. Lastly, we examine the evolution of functional traits and adaptations of Heteropogon and Thymida grasses in savannas and wetlands. First, to investigate phylogenetic patterns and relationships in Thymida and Heteropogon, so we use genome skimming data to assemble plastomes and pull out nuclear genes. Heteropogon and Thymida are polyphyletic genera, and all species are considered belonging to one clade, as it will be called Heteropogon and Thymida clade throughout this presentation. This coordinate pattern between plastome and nuclear genes suggests that possible past hybridization among species. Next, dating analysis from the plastome tree indicated that the diversification of Heteropogon Thymida clades occurred during the Miocene grassland expansion. There are three main events here. Heteropogon contortus diverged from the other Thymida at the beginning of the events, representing the oldest lineage of the clade. So lineage of Thymida arundinacea and Thymida villosa form a clade separated from the other Thymida species. The rest of Thymida species diversified later in the ecological time and even spanned into the Pleistocene. The spread of the two dominant species, Heteropogon contortus and Thymida triana, did not happen until the Pleistocene. The evolution of the clade occurred in a short period of time of less than 10 million years. The phylogeny was mapped with habitat preferences across the species. Habitat information was reviewed from ecological and taxonomic literature and summarized in this mapped phylogeny here. The pattern suggests that the habitat preferences were switched back and forth throughout the lineages, from wetland to savanna and from savanna to wetlands. Notice that wetland species are clustered within the sole Thymida lineage. These species are restricted in Australasian and Southeast Asian regions, where wetlands and savannas form mosaic habitats across the continents, and the proximity between these two habitats could promote species to migrate and adapt to the new environments. Bioclimatic characteristics were explored and verified using statistical tests on bioclimatic data extracted from species occurrences. PCA analysis separate savanna and wetland groups based on these four bioclimatic data: temperature seasonality, precipitation seasonality, mean annual precipitation, and precipitation of driest quarter. As you can see here, wetland species receive higher annual precipitation than savanna species. Lower precipitation seasonality and higher precipitation of driest quarter indicate that wetland habitats retain standing water during dry seasons. Bioclimatic evolution provides evidence that possible ancestral habitat resembles savanna environments. The mean annual precipitation was estimated to be intermediate, around 1,200 millimeters per year. The precipitation seasonality of 90 is equivalent to around 5 months of a dry season. Very low precipitation of driest quarter indicates that drought was present in the ancestral state. From this, it can be interpreted that Heteropogon Thymida clade originate in savanna and then migrate to wetland later in the time frame. This also supports that the biome shifts occur and could play as evolutionary drivers within the lineage. Differences in functional traits align well with differences in bioclimatic niche between savanna and wetland groups, which explain the adaptations in savanna and wetland species. Coordination analysis of vegetative traits confirm separation between savanna and wetland groups. All vegetative traits here are significantly higher in wetland species, especially leaf length and maximum heights. However, on length is not statistically different between savanna and wetland groups. Height and leaf length have been increased in wetland species in greater trait space. Heteropogon Thymida clade belong to tribe Andropogoneae that the ancestor had relatively tall stature and greater leaf length compared to other grass lineages. These provides opportunities to increase stature and leaf size. Increase in plant height and leaf length are more constrained than reducing the size of both traits. Increased height and leaf traits among wetland species imply active response of adaptive phenotypic plasticity as they are investing and maintaining growth in the presence of inter- and interspecific competitions under high availability of water. We found that the presence of straight-on is confined in wetland groups. At the same time, on types vary among savanna species, but most species possess geniculate-ons. Geniculate-on is ancestral state, and straight character and the absence of on are derived character. Different on types pointed out the ecological significance between savanna and wetland species. Although geniculate-ons respond to moisture and then move and twist, this mechanism is only effective when soil surface is accessible for seed burial, which facilitates seed establishment and fire escape when dry seasons are approaching. Imagine wetland conditions where standing water is maintained. Straight-on could give more benefit than the geniculate-ons, as straight column of the on could directly embed into the soil. In addition, the spores of wetland species could be dispersed by water, and at the same time some species lack of on at all. In conclusions, shifts between savannas and wetlands occur in heteropogone thymidoclade in labile fashion. Ancestral environments resemble savanna conditions, showing long dry seasons and droughts. This indicates that shifts into wetlands occur later in the lineage. Precipitation factors are important determinants in savannas and wetlands occupied by heteropogone thymidoclade. Functional traits of savanna and wetland species evolved through different trait spaces. Height and leaf length have been increased in wetland species and decreased in savanna species. On types are mostly geniculate-ons in savanna species and straight-ons in wetland species. Before I finish the presentation, I'd like to thank Dr. Michael R. McCain for providing DNA sequence data and Dr. Sophie Mancy for providing DNA samples. And I'd like to thank DPST for providing scholarship for my PhD project. I'd like to thank Kew Herbarium, Chinese Academy Herbarium, Forest Herbarium, Naturalist, Natural History Museum, Paris Museum, and EDB Lab in Toulouse. Thanks for your attention. If you have any questions or requests, please contact me through email or Twitter left in the first slide.